Welcome to Biomindscape. Today we are going to know about a new topic, that is the Hogsten base pairing. That is, we know that when we think of an DNA, we imagine a double helix structure which consists of a perfect base pair, adenine group with thymine, guanine, and cytosine. So all are neatly aligned through the hydrogen bond. So this classical image was given by Watson and Crick based on the base pairs rule described in 1953. But what if we told that the DNA doesn't always follow those rules? So DNA breaks its own symmetry to survive, adapt, and be recognized. So that is a Hogsten base pairing. It is a hidden and flexible phase of a DNA. So the classical base pairing, in this classical Watson and Crick base pairing, DNA bases align face to face, where adenine is paired with thymine through 2 hydrogen bond and guanine paired with cytosine through 3 hydrogen bond, and this geometry is planar, stable, and form the base of the famous B-form DNA. But this isn't the only way the DNA can function. Under certain conditions like DNA damage, protein binding, or chemical stress, base pair can change its geometry, and this is where the Hogsten base pairing comes into play. So in 1963, the scientists cast Hogsten observed something unexpected using a X-ray crystallography. So he found that adenine and thymine could pair in different way. One where adenine used its N7 position instead of N1 to form a hydrogen bond with the thymine's N3. So this alternative geometry was later named as Hogsten base pairing in his honor. So what is the difference in Hogsten pairing? In Watson and Crick pairing, both base remain an anti-conformation. Adenine uses its N1 atom to hydrogen bond with the thymine N3. So the base pair lies flat and maintain the double helix structure. But in Hogsten pairing, the purine base, it can be adenine or guanine. It flips into the syn conformation, where the adenine now use N7 and sometimes C6 to bond with the thymine N3. So this rotation changes the shape and hydrogen bonding pattern, and this base pair is now it is tilt and not a flat. So this reveals the Hogsten edge of the purine and a side usually hidden in the standard double helix. So here AT Hogsten pairing, it is seen under the DNA stress, or whereas there is methylation, or when the DNA binding proteins are there and adenine flips in those syn and form a bond using N7 and N3. This is about the AT Hogsten pairing, whereas the GC Hogsten pairing, it is common in triplex DNA and at lower pH, where cytosine gets protonated, that is C plus, and guanine use N7 and cytosine use N3. So the main feature is base conformation of Hogsten, where syn, that is a purine, and anti, that is the pyrimidine, is there, and hydrogen bond is facing the Hogsten edges, the major groove, then the groove exposure, major groove, more exposure and stability. They are less stable but more flexible, and they are found in the triple X DNA, protein DNA, and lesion. So where does this Hogsten pairing occurs? In the triplex DNA, also known as the HDNA, where a third strand binds to the duplex DNA using a Hogsten interaction, important in the gene regulation and chromatin remodeling. Then G quadruplexes, where the 4 guanine form a square planar structure using Hogsten hydrogen bond. They are found in the telomeres, oncogenes, and regulatory DNA. And the DNA protein complexes, in that also the transcription factor like P53, TBP, and polymerase induces local Hogsten pairing to recognize and bend the DNA. So DNA damage response, in that also the Hogsten pairing help in tolerating the lesion such as the methylated guanine or oxidative damage by maintaining the pairing under distortion. Then it have some medical and technological significance. So understanding Hogsten base pairing isn't just academic, it is a real world application, whether drug targeting, gene therapy, and epigenetics. In all this, it is have some important significance. In drug targeting, anti-cancer and an antiviral drug can be designed to bind the DNA region that adopt the Hogsten geometry. And gene therapy also, the triplex forming oligonucleotide use Hogsten pairing to silence the specific gene. In epigenetics, the DNA methylation can promote Hogsten translation and influencing the gene expression. So Hogsten pairing, base pairing, which atom of adenine form the hydrogen bond with thymine? That is the N7 bond. So to sum up with the Hogsten base pairing, they are the hidden features of DNA that allow it to bend, flex and adapt, and through less stable than Watson and Crick pairing, they are versatile during the stress protein interaction and DNA damage. They remind us that DNA is not just a rigid ladder, it is a dynamic, intelligent molecule capable of switching from to meet the needs of the life. So next time you imagine a double helix, remember there is more than one way to the pair of this base. So if you found it helpful, don't forget to like, share and subscribe and hit the bell icon. Stay tuned for more deep science explanation.